Okay, folks, so I restarted the movie because I realized a lot of it was recording and there was nothing happening. So let me just do a quick recap, you guys. So for the movie, this is Tuesday, October 22nd, 2019. With regard to announcements, folks, there's one handout. There's a new handout. And this handout, you guys, has the bonus topics for Lab Exam 2. And the bonus topics will be the Enterobius vermicularis, common name pinworm. And did we talk about the Scotch tape prep, you guys? Yeah, okay, so Scotch tape prep. So you know on the lab exam, if I say, here's a photomicrograph of eggs that were acquired around the anus of a child with an itchy butt, probably more scientific term than that, right? And if we say Scotch tape test, right, or we collected the eggs with Scotch tape, you know it's got to be who? The pinworms, right? Because again, the females come out at night and they lay their eggs around the anus, right? And it causes an intense itching sensation, right? So especially with little kids, if you see that they're scratching at their bottoms all the time, think about pinworms. Okay. And then, folks, there were two helminths that are common parasites of dogs and cats. So the next one down there, you guys, is the Dipylidium caninum. This is the common tapeworm of both dogs and cats, right? Rarely is it zoonotic. What does that mean? Rarely do we get infected, right? So it's mostly just a concern of your dogs and cats. And we want to remember, folks, with the dog and cat tapeworm, an intermediate host are fleas. So if your dogs or cats have tapeworms, not only do you want to go to the vet and get medication to kill the tapeworms, but you also need to get flea control, right? Because as long as the fleas are around and if they're carrying the larva, right, your dogs and cats will get reinfected. And then, folks, the next one there is the dog heartworm, Dirofilaria immitis. We have it here in Northern California. So if you have dogs and probably you have your dog on heartworm preventive medication, right? And just like with Wuchereria bancrofti, lymphatic filariasis of humans, the vector of the dog heartworm is what? Mosquitoes, right? Mosquitoes. Good job, you guys. And then the last one, I'm not sure, did we mention the flea-borne typhus, the outbreaks they've had down in Southern California? Had we mentioned that last time, folks? Right, okay. So that would be another bonus topic. And remember, with flea-borne typhus, it is a bacterial pathogen, Rickettsia typhi, so it can be treated with antibiotics. There are reservoirs of, like, rats are common reservoir hosts. I think on the handout we had, there was, like, raccoons, opossums, maybe even cats. So the non-human animals get infected, the fleas bite them, and then subsequently, if those fleas bite us, they inject us with the Rickettsia typhi. And we said that in spring they had an outbreak down in the LA city government offices. Apparently they had a rat infestation. So the rats brought the infected fleas in, and then the fleas bought the employees, and they developed typhus. So one thing they were doing, they were pulling up the carpet, right? Because I guess a lot of the fleas were hiding in the carpet, right? Yeah. And then we also said, folks, also of concern are our community members that are homeless, because often they're at high risk for example, the flea-borne typhus. Yeah. Okay, so those were our bonus topics for Lab Exam 2. And with regard to Lab Exam 2, folks, remember that there were study guide questions at the end of the helminth vector handout, right? So you want to do those study guide questions. And then we passed out a multi-page packet of new lab homework study guide questions, right? And I ordered them by the sequence in the lab manual, folks. So you're like, Chapter 9, motility, what? So if you turn the page to page 2, you'll see that's where Chapter 10, Microbial Growth Media, starts. And there's quite a few homework study guide questions there. So again, you want to start working on those early.
So when you turn this in, just go ahead and disregard the
Oh, you're not going to disregard this. It's just that we're going to do Chapter 9 later. We do the chapters out of sequence.
Okay.
Go figure, right? Okay. But just like with Lab Exam 1, folks, you'll be turning in the vector helminth study guide questions and this packet. And again, that'll be worth 20 points. And you'll be turning it in on the day of the lab exam. All right? Okay. let's see here. And just other announcements, folks. So we're going to have Open Lab this Friday from 9 a.m. to 11 a.m. And Open Lab is for both lecture and lab questions. So because we have lecture exam 2 a week from today, right, and it's a tough one, you guys. Metabolism, microbial growth, microbial genetics. The Open Lab would be not only to talk about lab questions, but also if you want to come in and talk about any lecture exam 2 questions, okay? And also, folks, after we do our introduction, I'm going to be passing back the lab exams. And folks, the overall average for all 4 lab sections was 84, which was just amazing. Back in the day, we would get averages of like 60%. So you guys are doing really well, really proud of you. And the Tuesday, Thursday lab sections had the highest averages on the multiple choice part. I didn't separate out total lab exam scores based on labs, but the scoring machine told me that the Tuesday, Thursday lab sections had the highest averages on the multiple choice section, which was 90%. It's like, well done. That's really good, you guys. Whatever you're doing, keep it up because you're doing a good job. And furthermore, folks, just really quickly, if you didn't do that well, remember all that goes into the point spreadsheet are just points. So if you had a bad day, if you had a bad lab exam, we have over 1,000 points in the course. So don't feel like, oh my gosh, I'm going to fail the course. There's enough points where you can have kind of a rough exam and still do okay. And also remember, folks, that on the day of lecture exam 3, you can turn in 2 little mini extra credit posters, and each of those is worth a maximum of 5 points. So in theory, you could say, okay, right there, I've got 10 points right there, right? And so you might say, well, maybe I didn't do so good on this lab exam, but I'll do the 2 extra credit posters, and that'll be an additional 10 points, right? So remember, you guys, just don't let one not so good exam discourage you, right? Be persistent, be stubborn. Okay, all right. Okay, and let's see here, folks. So, do we have our lids off right now, you guys? Because it would be really good, because this intro is a little bit lengthy. So all the teams have the lids off, and somebody's timing. Okay, so if the timer goes off, I'll try to stop to give you a break to get your lids back on. Okay, all right. So, folks, let's take a look at our Chapter 10 Microbial Growth Media handout. This was developed by a fantastic micro tutor we had a few years ago, Audrey Fung. Audrey Fung subsequently went to pharmacy school in Hawaii. We said, we all need to come with you, Audrey, to pharmacy school in Hawaii. And she's now graduated. And I'm not sure where she's practicing at this point, but she was really a wonderful, dynamic micro tutor. And she did this study guide. I'll show you a study guide she made for metabolic tests, and she also did a study guide for immunology and lecture. So you'll be seeing more of her study guides. So folks, let's use the study guide just to kind of direct our discussion, and I'll ask some questions. Now, and I know sometimes, folks, it's hard to be looking at 2 things at once, but what I noticed yesterday, some folks were looking at their Chapter 10 Microbial Growth study guide questions, and if we hit one of the questions, they were writing down the answer. And sometimes that's hard because you're like flipping back and forth, but it's one strategy that some folks use. All right, folks, so first of all, let's just ask ourselves, what do we mean by medium or media? So it's just food for our microbes. Medium, singular, media is plural. I'll never mark you off if you get singular and plural mixed up on an exam. Right, so it's food for our microbes, because very often in our microbiology lab, we're trying to get our microbes to grow, to replicate. And this might be because we're trying to diagnose maybe someone's suffering from an infectious disease, or we're trying to figure out which microbe it is. Maybe we're public health microbiologists and there's been an outbreak of fecal-borne pathogens in our community, and we're trying to figure out what's the source. Is it water? Is it food? Right? We need to grow our microbes to do metabolic tests. We need to grow our microbes to do antibiotic sensitivity testing. These are just a few examples of why we want to grow our microbes, feed them, have them replicate. Okay, so if we're going to talk about, folks, different types of media, And you can see there's different ways to classify media. So the simplest way is to classify the media based on the physical form it takes. So if we have a liquid media, what would we call liquid media? What would we call liquid media, folks? Broth, yeah. And just think of like chicken broth, like chicken soup. That's what I think of. Broth, it's like a soup for our microbes. And just like we eat soup, right, to get nutrients, we can make a soup or broth for our microbes to feed them so that they can grow. Now, folks, if I told you that this broth had been autoclaved, so it should be sterile, just eyeballing it, folks, do you think I'm right or am I pulling your leg? Does it look sterile? Yeah, you're right. I agree. And why? Why did you make that conclusion?
It's not too cloudy.
That's right. It's not cloudy. It's transparent. So, folks, we use cloudiness, and the fancy term is turbidity, is a sign that cells are present. So let me see if I can find a cloudy broth in this selection. Let's take a peek here. I thought we had one yesterday. Okay, so if we have cloudiness in our broth, turbidity, we know cells are present. Can we know with certainty, folks, if the cells are alive or dead? No, we won't be able to know that, right? This is an example of turbidity. See how cloudy it is, right? So folks, if you're growing cultures in broths, right, you put them in an incubator, when you pull them out, just gently mix them. Sometimes the cells will settle to the bottom, and you might think, oh, they didn't grow, right, because the broth appears transparent. But again, just gently tap them so that it will resuspend the cells. Okay. So again, folks, we know cells are present. We don't know if they're alive or dead. Can we tell, folks, just looking at the broth culture, is this what we call a pure culture, meaning a single type of microbe? Can we tell just by looking at it? No, we can't. So that's a disadvantage of broth cultures, right? We can't tell if it's one microbe or it could be 10 different microbes in there, right? We can't tell just with our naked eye. Good. Okay, folks, and then if we add a solidifying agent to our broth, right, we'll make solid media. And the most common solidifying agent we add to our broths to make them solid is an extract of seaweed called agar. And the reason we use agar is that most microbes can't hydrolyze it. Most microbes don't have the enzymes to break it down. If they did, they would convert our solid media back to a liquid media, right? We just have this soupy gamish. So it's good that most microbes can't break down agar. And furthermore, folks, it's really important because on a hot Sacramento day, once the agar is solidified, it won't melt on a hot Sacramento day, and it won't melt when we put it in the incubator. One of the first solidifying agents that was used in microbiology was gelatin, an animal protein, like jello gelatin, right? But disadvantage, a lot of microbes make enzymes to break down gelatin to use it for food, right? And then it liquefies. And also it will melt on a hot day, right? So this is why eventually agar became the primary solidifying agent used in media. So folks, if we put our agar into a tube, and after autoclaving, let the agar solidify at an angle, what do we call this? Agar slant, good. And if we let the agar solidify with the tubes upright, this is called an agar deep, right? These are nice for trying to see if our microbes can grow anaerobically in the butt. Good. And folks, if we pour the agar into a petri dish and let it solidify, what do we have? An agar plate. Good. Good job. All right. Now, the advantage, folks, of agar, and for me specifically agar plates, folks, is that first of all, we can see if microbes have grown. How do we know microbes have grown, you guys? So compare this TSA plate to this one. How do you know microbes have grown on this plate? You can see the colonies, right? You can see the colonies, yeah? So that's how we're going to determine. And it's really helpful, folks, because as an example, today when I was taking the TSA plates out of their sleeves, they were supposed to be sterile, but I found this one. How do I know this one was not sterile? Yeah, see, we have, oops, it's dripping down on my sleeve. Okay, let me go take care of this. But we can see right off the bat, right, that this plate wasn't sterile. So let me decontaminate myself here. Okay, good. And folks, if we saw 10 different qualities growing on our plate, do you think think that's a single type of microbe, or could it be 10 different microbes? Yeah, it could be 10 different microbes. So another advantage of growing our microbes on our agar plates is we can get an idea of how many microbes landed on the surface initially, and we can get a rough idea of the variety of microbes that are growing. And that's something that raw cultures don't permit us to do. Okay. And furthermore, folks, is an isolated colony considered a pure culture? Yeah, because again, the hypothesis is that an isolated colony arose from a single cell landing on the agar and then them growing and dividing, growing and dividing, growing and dividing. So indeed, the cells within a colony would be, we would consider them genetic clones of one another. Good, okay. All right, folks. So let us turn the page. All right, and we're gonna, okay, so another point here, folks, is in our lab, right, let's say, folks, we're talking about our plates. Can we grow in our lab strict or obligate anaerobes, say, on our plates? What do you think? The microbes growing on the surface of your agar, do you think they could be strict or obligate anaerobes if they're growing on the surface of your agar? No, right? Because what's present on the surface of the agar? Oxygen, right? So you guys, in our lab, we don't have the facilities to grow strict or obligate anaerobes, right? In well-equipped microbiology labs, you'd have an anaerobic hood, right, where everything is kept under anaerobic conditions. So when you're processing your samples, they're never even exposed to molecular oxygen. So in our lab, we aren't growing any obligate or strict anaerobes. We can grow some facultative anaerobes, aerotolerant anaerobes, but no strict or obligate anaerobes. Good. Okay, folks, now again we're on page 2, and it says classifying media by its functional function or functional type. And this is a classic lab exam question where we'll have at a station different types of media that we'll be talking about this week, and we'll ask you to classify them. All right, so let's use some of the terms. So folks, all-purpose media is media that can support the growth of a wide range of bacteria and fungi, right? So the all-purpose medium we're using today is the tryptic soil agar, right? So we're going to predict that you guys will be growing both bacteria and fungi on your TSA, your all-purpose medium, and the fungi would include both molds and yeasts, okay? Then folks, enrichment media. Enrichment media, we take a TSA, an all-purpose medium, and we're literally going to add blood to it. So how do you think, folks, blood agar appears? In contrast, folks, to the TSA, what color is the blood agar? It's red. Why is it red? Why is blood agar red? Does it have red blood cells in it? Erythrocytes, right? And what color is hemoglobin when it's oxygenated? Bright red, yeah, right? So you guys, you might say, well, why would you add blood to an all-purpose medium like TSA? Well, the reason is that many of the pathogens that infect your patients or infect other animals, they're what we call fastidious pathogens. And fastidious pathogens can't make a lot of things by themselves. We say they have need for growth factors, so they need pre-made amino acids, pre-made precursors to nucleotides. They need lots of vitamins, right? So do you think, folks, that blood would be a wonderful source of preformed organic molecules and growth factors? Yes, blood is wonderful, right? So we want to make sure in the microbe lab that we give our fastidious pathogens all the things they need to grow, right? Because if they can't use the food or the food is insufficient, they won't grow, and you could end up with what we call a false negative, right? So definitely in microbiology diagnostic labs, when you receive a sample, you're always going to put some of the sample onto blood agar, right? Because you don't want to miss those fastidious bacterial pathogens. We're also going to see, folks, that in addition to being an enrichment media, blood agar is also what we call a differential media. and we're going to come back and talk about why blood agar is differential. Okay. So now, folks, we're going down to the next one, selective. So selective media is media that includes some substance or substances that inhibit, stop the growth of certain groups of microbes, and thus permit or selects for the growth of other microbes. It's a little bit confusing initially. So, for example, folks, let's look at the Sabouraud's dextrose media that we're using. Sabouraud's dextrose media is selective for fungi. Okay, so on your sab plates, we should see primarily yeasts and molds growing. Now, why are the bacteria inhibited? And it's because the Sabouraud's dextrose medium, it has a pretty low pH, pH 5.6. Most environmental bacteria don't grow very well at that low pH, but most environmental fungi do. They can grow at a little bit lower pH. And the second reason is that they add a really high concentration of dextrose. Dextrose is the same thing as glucose. Historically, I don't know why that they say dextrose and glucose. It seems to me usually in medical facilities they call glucose dextrose, and usually in lab facilities they call glucose. But anyway, so glucose and dextrose are the same thing. So that high concentration of dextrose, glucose, can also help inhibit the growth of a lot of environmental bacteria. And again, the fungi are pretty comfortable growing at high glucose, dextrose concentrations. Okay, so you guys, what would be your guess? Looking at total number of colonies, which medium do you think should grow the greatest number of colonies? Our all-purpose DSA or the selective SAB? The all-purpose, right? Because we should be growing both bacteria and fungi, right? On the SAB, we're going to think most of them are going to be fungi, either yeasts or molds. Good job, folks. All right, and then, folks, under selective, there's this little table. So SAB was down at the bottom. Now let's take a look at this mannitol salt agar, right? This is another example of a selective media. And the inhibitor in mannitol salt agar, you guys, what is the inhibitor? And you can read it right off your chart. 7.5% sodium chloride, right? Good old table salt. And folks, this is often a lab exam question. So I'd like you to know, for mannitol salt agar, the inhibitor is 7.5%, and this is weight per volume of sodium chloride. So I'd like you to know what that means, that 7.5% weight per volume. So that means that it's 7.5 grams of sodium chloride in every 100 mils of media. So folks, if I ask you to make 1 liter of MSA, how many grams of sodium chloride are you going to add? So 1 liter is 1,000 mils, right? So how many 100 mils are in 1,000? 10, right? 10 times 100 is 1,000. So you guys, how many grams of sodium chloride would you add to a liter of MSA? 75 grams. Good. Okay, and that's kind of a classic lab exam 2 question. Now you're like, well, who cares? Why does that make the medium selective? Well, that's a really high salt concentration. Most microbes can't grow at such a high salt concentration. But folks, can you think of a place in nature that's pretty salty, like maybe on our bodies? Our skin, right? Our sweat has a pretty high sodium chloride concentration for evaporative cooling. It's also inhibits the growth of microbes, right? So folks, do you think through natural selection, microbes that have evolved to live on skin, do you think they could grow at pretty high salt concentrations? Yeah, they can. And there's a really important bacterial pathogen, you guys, that does live on our skin, and that's the pathogen Staphylococcus aureus. So salt tolerant Staphylococcus aureus. Do you think Staphylococcus aureus maybe can grow on mannitol salt agar? Yeah, it can. And indeed, folks, mannitol salt agar, we say it selects for salt-tolerant microbes such as members of the genus Staphylococcus, including Staphylococcus aureus, right? So One example might be, folks, maybe your patient has an abscess, right? And maybe it's popped. So you take a swab and try to harvest some of the pus from within, right? And your big concern is Staph aureus, right? So you know you're probably picking up some other microbes, right? But you're specifically trying to see if Staph aureus is growing. Could you take some of the pus and inoculate a mannitol salt agar plate and incubate it? Yeah, and if Staph aureus is present, are you going to get good growth? Yeah, you are. So that's like an initial step, right, in checking for the presence of Staph aureus, say in an abscess. Good. Oh, that's great. We should do that. Every lab, every lecture, we should have a little break. Okay, I know that might be ugly if I'm up here dancing. Okay, we won't do that. So you guys, you need a minute or two just to put the lids on your plates. Okay. All right. So you guys, if I asked you to classify Sabouraud's dextrose agar, is it all-purpose, enrichment, selective? So you guys, so pretend it's a lab exam. Classify by functional type Sabouraud's dextrose medium. Is it all-purpose, enrichment, or selective? Selective. It's selective for fungi, right, such as yeast and molds. What are the inhibitors? Okay, high concentration of glucose dextrose, and what else? Low pH, right, low pH. Good job, you guys. Mannitol salt agar, is it all-purpose, enrichment, or selective? Mannitol salt agar? It's selective, right. What does it select for? Salt-tolerant microbes, such as members of the genus Staphylococcus, and we're really looking for which pathogen? Staphylococcus aureus. Good job, you guys. All right. Folks, classify tryptic soy agar. All-purpose, good. We would expect to grow what? Good. Wide range of bacteria and fungi. Good job, you guys. Classify blood agar. Enrichment, right? Why is it red? Yeah, whole blood or wash red blood cells. Good, you guys. Why do we want to use blood agar when we're looking for human or animal pathogens? A lot of them are fastidious, right? They need those preformed growth factors, amino acids, nucleotide precursors, and vitamins. Good job, you guys. Excellent. Now, folks, on the selective chart, it mentions another medium called McConkey medium, or McConkey's agar. We're going to talk about that on Thursday, right? And so what we're doing, again, guys, we're kind of taking the onion approach. We're peeling back the first layer of our onion, and then we're going to go deeper on Thursday when we'll actually have some demo plates for you of selective and differential media. What I wanted to finish with is just a really brief discussion, you guys, of what's called differential media. And I'm going to give you one example today, and then I'll give you more examples on Thursday, okay? So differential media, folks, let me see where Audrey has that. Yeah, okay, so you guys, if we keep going down to the bottom of page 2, it says they're differential. So differential media lets us distinguish between different groups of microbes that are growing on a particular medium. I love it. I love differential media because it'll change appearance after incubation, and I don't know, I'm just totally entertained by that. So the example we're going to use today, folks, is going to be the blood agar. And blood agar is differential based on a phenomenon called hemolysis. So differential, so we're going to use as our example today, you guys, blood agar. Okay, so we know that blood agar, it's enrichment, right? And it's also differential. So we'll see Thursday that media can have more than one function. So you guys, let's take a look at blood agar then. Okay, so it's enrichment and what else? And differential. Good job, folks. And it's differential based on which property? Based on hemolysis, hemolysis. What do you think hemolysis is? What does hemo refer to? Blood, right? Blood lysing. Okay, so hemolysis is the ability to break open red blood cells, and it will change the appearance of the agar, folks. So let me get let me get the plate that we did yesterday. So, and I'll do the same thing today, folks. I asked for 4 volunteers, and from 2 of your colleagues, we did throat swabs, and from 2 of your colleagues, we did nasal swabs. Blood agar is so much fun, you guys. So, we'll do the same thing today. We'll ask for 4 volunteers. You see, you guys, this is the fresh blood agar. This is after incubation with throat and nasal swabs. Do you see a difference in coloration of the agar? Yeah, so this is the hemolysis we're talking about. So let's do the hemolysis, folks. So there's 3 types of hemolysis. And again, folks, the type of hemolysis is a clue to the identity of the microbe, right? It can be really helpful. So the first type of homologous you guys we're going to talk about is called alpha. So we use the alpha symbol. So alpha hemolysis. And let's, again, folks, let's use Audrey's handout. And let's see here, the discussion, folks, of the hemolysis is going to be on page 1, 2, 3, 4. And folks, this handout is on Canvas and it's in color. So it's going to be such a helpful study guide when you get ready for lab exam 2. So guys, so page 4 it says blood agar. So the first type of hemolysis, you guys, is what? Alpha hemolysis, right? And this is sometimes described as, it sounds kind of goofy, partial hemolysis. It's like, what do you mean by that? So Audrey has a really good description. So partial hemolysis, we're getting lysis of the red blood cells, but the contents are not being destroyed. The contents are not being destroyed, and as a result, we get this greenish, dark discoloration of the agar surrounding the colony. So you guys, can you see here how on the throat swabs especially, it looks kind of green? That's alpha hemolysis. That's alpha hemolysis, right? So we can describe it as a partial hemolysis. The red blood cells are lyse, they're broken open, but the contents aren't destroyed, right? And so Audrey did the research and found out that it's the hemoglobin when it gets oxidized. I'm going to have to check that out. Is it oxygenated versus oxidized? Okay, it's the hemoglobin that's giving that greenish brown color. Okay, so we would describe this as a darkening or greenish discoloration of the agar. And for the lab exam, you guys, I'd like you to know 2 alpha hemolytic bacteria, and this will make sense why the throat swab has a lot of alpha hemolytic colonies, because the oral microbe, Streptococcus mutans, is alpha hemolytic. Folks, why did we discuss Streptococcus mutans? What is it involved in? Yeah, the tooth biofilm, right? They make the slime layers, they stick to our teeth, and they ferment sugar, make acids, decalcify our teeth. So this is why, you guys, if you do a throat swab, or you collect saliva, or if you were to swab your gums, and then transfer your specimen to blood agar and incubate it, you're going to get nice alpha hemolysis, yeah? A second pathogen I'd like you to know is another member of the genus Streptococcus, and this is Streptococcus pneumoniae. What do you think Streptococcus pneumoniae causes? Yeah, pneumonia, right? This can be really serious in older folks, right? And in some cases, it can invade the bloodstream and even invade the central nervous system. So again, in your older patients, Streptococcus pneumoniae can be a really serious pathogen. Okay, guys. So, folks, what's the second type of hemolysis that Audrey has described here? Beta hemolysis. So we use the beta symbol. And beta hemolysis, you guys, is described as complete hemolysis, meaning not only are we going to break open the red blood cells, we're going to destroy the contents. So it's just wild, you guys. The agar becomes clear as glass. And a really good beta hemolytic colony, you could lay the plate down on top of your textbook and you could read through the area of beta hemolysis. That's classic, right? So it's complete clearing. It's actually clearing of the agar, clear as glass. And there are 3 really important bacterial pathogens that I'd like you to know that are beta hemolytic. And we might, I'm not sure, we might have to do some follow-up here. We might have one of them on this plate. And you guys, I know this is going to be hard to see because we don't want to open these, right? Because we're amplifying pathogens now. But on this one throat sample, if I backlight it, can you see on this one throat sample how there's areas that are extreme clearing almost? I mean, you see the greening, right? But you see how the agar is even lighter there? That might be beta hemolysis. And if we're getting beta hemolytic colonies out of a throat swab, our prime suspect is Streptococcus. Anybody you want to guess? Pyogenes, literally means pus producer, right? So if you had a patient with a sore throat, right, one of the things you always want to think about is Streptococcus Pyogenes. So you can take a throat swab, and if you have your own little micro lab, right, you'd streak it on blood agar, incubate it, and the next day you'd see these little tiny colonies that are beta hemolytic. And I'm thinking we might have some Streptococcus Pyogenes here. Now, it's not a surprise, folks, because as adults, probably 10-15% of us carry Streptococcus Pyogenes in our throat without any clinical signs or symptoms, right? So we have a really high symptomless carrier rate. Now, what I'll do is I'll ask your colleague whose throat sample this was if they have a sore throat. And if they have a sore throat, I'm going to recommend they go to the doctor because it might be Strep Pyogenes. And with Strep Pyogenes, you guys, you actually do want to treat with antibiotics because there are some immune sequelae that some strains will cause. They can cause like heart damage, kidney damage. So if you have a strep throat, I mean, I really would encourage that you go to your healthcare provider and get antibiotics. Have them diagnose it and get antibiotics for it, right? Because you don't want those immune sequelae to it. All right. And then, folks, another pathogenic member of the genus Streptococcus. Do you see so far we've got 3 members of the genus Streptococcus that are pathogens, right? So a lot of pathogens in Streptococcus. They're gram-positive bacteria in chains, so we can treat with antibiotics, right? This next one, you guys, is called Group B strep, or Streptococcus. Group B Streptococcus. And often in textbooks, you'll see it as GBS. And this has kind of an interesting history. Up until about 30 years ago, everybody thought this bacterial pathogen was just a pathogen of like farm animals, right? It would often invade the mammary glands of cows, causing mastitis. But everybody thought it was just a veterinary pathogen. And in vet medicine, you guys, this one, you don't have to remember this, but in vet medicine, we know it by its scientific name, Streptococcus agalactia. And the reason it's called agalactia is it can decrease milk production in dairy animals, right? But again, folks, in human medicine, probably no one will know the scientific name. It's just in vet medicine. So what happened about 30 to 40 years ago, Group B strep became an emerging disease of humans, right? Like a new disease of humans. And the heartbreak is, you guys, is that in pregnant moms that are colonized, so they're colonized in the vaginal mucosa. If they deliver their baby, the baby can become infected. And the Group B strep can cause neonatal sepsis and meningitis. Neonatal means newborn. Neonatal sepsis, so for example, the bacteria growing in the bloodstream, and we can get invasion of the central nervous system, and it can cause meningitis. And it can be lethal to little babies. So about 30 or 40 years ago, they weren't screening women for colonization until they started identifying this was a pathogen killing newborn babies. So now, if you're pregnant, you should definitely be screened for Group B strep. And you guys, it's not a problem. It's sensitive to antibiotics. So they just put them all on antibiotics, maybe screen again right before it's time to deliver the baby. But as long as you know ahead of time that you can treat with antibiotics, it doesn't have to be lethal for the baby, but it is important that you're screened. Okay. All right, folks, and then the last one, the last beta hemolytic, is not a member of the genus Streptococcus. It's our not so good friend Staphylococcus aureus. And again, folks, a lot of us carry Staph aureus, especially in our nose, right? So this is of concern, say, in hospitals where the medical staff are working with immunocompromised patients or, say, burn patients, because you can be a source of the Staph aureus. So sometimes what they'll do is they'll do screening, right? They'll take nasal swabs, and you again, folks, you inoculate your blood agar, and then you look for beta hemolytic colonies. Now, I don't see any beta hemolytic colonies in the nose swab of your two colleagues, right? So it doesn't look like they're carrying Staph aureus. I've done it on myself before, and I'll get Staph aureus out of my nose. So I know none of you are going to want to shake my hands now. But anyway, okay? All right, so you guys, can you name 3 bacterial pathogens that are beta hemolytic? Streptococcus Pyogenes, and what does it classically cause? The Streptococcus sore throat, right? The so-called strep throat. Okay. Can you name another member of the genus Streptococcus that is beta hemolytic? Group B strep. Now, who are you really worried about? Pregnant patients. Why are you worried about your pregnant mamas? Yeah, because if colonized, the baby can become infected, and it can cause what? Neonatal sepsis and meningitis. Good. And folks, what about a member of the genus Staphylococcus that is beta hemolytic? Staphylococcus aureus, okay. And where's a really common site where we can carry Staph aureus? In the nose. Good. Good job, folks. Excellent. And then the last type of hemolysis, you guys, I do not know why they named it hemolysis. It's called gamma hemolysis. And it's no hemolysis. It's like, that's silly. Why would you name it gamma hemolysis when there's no hemolysis? Oh well. So folks, most microbes are gamma hemolytic, right? So hemolysis, the ability to cause lysis of red blood cells, it's a pretty unusual property, right? So most microbes are going to be what? Gamma hemolytic, and how would that appear? No change in the agar, right? No lysis of the blood cells. Good. You guys are awesome. Okay, so the plan, you guys, is on Thursday, we're going to do McConkey's medium, which is both selective and differential. And we're going to come back and talk about how mannitol salt agar is also differential, right? So we're going to see some of these media, they can have more than one function. Okay? So, I know, folks, what I wanted to do, 2 more things I wanted to do. I wanted to see if we could have 4 volunteers. And what we want to do is we're going to go into the hallway and have 2 do throat swabs. And 2 folks do nasal swabs. And what we'll do is we're going to, we'll glove up, right? And we'll go out in the hallway, and I'll give you the sterile swab, and you will do the swab yourself and then hand it to me. And then I'm just going to do, just like we did last time, you guys, I'm just going to make a quadrants here. And it's all anonymous. We just use numbers, so nobody needs to feel, anyway. Okay, and then we'll do just as we did yesterday. So we'll check for the alpha hemolysis and beta hemolysis. And again, you guys, if you have a sore throat and we have beta hemolysis, I know one of the folks yesterday had a sore throat, and I just don't remember which throat swab, but if that person with a sore throat is the one with the beta hemolytic colonies, I'm going to suggest that he or she goes visit their healthcare provider, because it could be strep throat. And that also means, you guys, we have to be really careful with our hands, right? This time of year, there's a lot of illnesses going on. Try to keep your hands away from your face, from your eyes, your nose, your mouth. And whenever you can, you guys just wash your hands, right? That's the best way. One of the most powerful ways not to get sick is to keep your hands away from your face and wash your hands. Okay, so after we do the throat and nasal swabs, you guys, I will bring in your lab exam 1, right? And I'll go over the the points so you can find out if I made a mistake. Let me know if I made a mistake that wasn't in your favor. If I made a mistake that's in your favor, just keep it. Don't even worry about it. Okay? So are there 4 folks that would volunteer? Awesome, Jessica. Yeah. Okay, great. Do we have 3 other folks? Yay, Jessica. 2 more brave souls? Great. Sarah, thank you. 1 more? You wanna? Yay. Okay, so you guys, let me stop the movie. And then the volunteers, if you guys can wash your hands, and then we'll go into the hallway and we'll glove up